That's what the federal taxpayer funding debate is about right now. It's not about these historic cell lines. Those lines were derived in the 60s and 70s from an abortion. But the cells that have been kept in culture for decades now are not fetal tissue. They're cell lines. And that's not under debate. There are actually better ways to make vaccines now than through those historic cell lines. Some examples include the new Shingrix vaccine for shingles, which is made in a non-fetal type of cell and is actually better than the earlier shingles vaccine made in fetal tissue. Another example is the Ebola vaccine, which now has been applied over 40,000 doses in this recent outbreak of Ebola in Africa successfully. The earlier study showed 100% protection of this non-fetal derived vaccine. The use of organoids, as my colleague has indicated, have also been used to determine the mechanism of action of Zika virus on the developing brains that results in that microcephaly we saw. Mr. Chairman, there are many other points we could make, but in summary, I want to say that again, there is no scientific necessity for continued use of fetal tissue. It presents no advantage to medical research. Instead, we have modern, advanced science that can be deployed more successfully and give better answers. Taxpayer funds should be redirected to the numerous modern, productive scientific alternatives. Thank you.
Thank you, Dr. Prentice. Dr. Temple, you're recommended.
Thank you. Chairman Meadows and Jordan and Ranking Member Krishnamurthy, thank you for the opportunity to testify at this hearing on the value of fetal tissue research. I am Scientific Director of the Neural Stem Cell Institute and here today on behalf of the International Society for Stem Cell Research, where I served as President. I offer my perspective as representative of nearly 4,000 research colleagues around the world and as a scientist who has spent 32 years pursuing research to fight human disease. Fetal tissue is an essential resource for study and developing therapies for cancer, HIV, Zika, tuberculosis, and other devastating diseases. Fetal tissues have unique properties. The alternatives mentioned may be useful at times, but cannot fully replace fetal tissue. Zika. Fetal tissue is key for studying developmental conditions, including birth defects associated with Zika. Through studying donated fetal tissue, we're learning how Zika infects specific types of brain cells, producing malformations such as microcephaly that can severely impact quality and length of life. This research would not have been possible with other research models because we cannot mimic normal fetal brain tissue using current stem cell technology, including brain organoids. HIV/AIDS. HIV is a human pathogen that attacks the immune system. Animals that model the human immune system are essential for HIV research. The development of Truvada, an antiretroviral therapy that prevents the transmission of HIV, depended on fetal tissue. These animal models, mimicking the human system, are also crucial for ongoing drug discovery for dengue, influenza, Ebola, tuberculosis, and other infectious diseases. The alternatives mentioned to this fetal tissue model are not enough. The so-called neothigh mouse that uses the surplus thymus does not model the full and long-term development of human blood cells, including the immune cells that are relevant to viruses like Zika and HIV. Cancer. Fetal tissue research is crucial for understanding why and how many childhood cancers develop. Recent research compared the stages of fetal retinal development to samples from children's eye cancers to identify the genes responsible for tumor formation and growth, opening new possibilities for drug discovery. Similarly, fetal tissue is helping scientists target genetic mutations responsible for children's muscle cancers. CAR-T immunotherapy, which has revolutionized cancer therapy, was made possible after decades of fetal tissue research. The development of these products would not have been possible without testing them in mice that closely model the human immune system. Our ability to develop the next generation of immune therapies for other types of cancer depends on fetal tissue-derived mouse models. Alternatives to fetal tissue are not sufficient. All of them come with significant deficiencies. Tissues from miscarriages frequently have abnormalities. Fetal cell lines are typically cancer-like and often composed of a single cell type rather than the many cell types present in fetal tissue. iPS-derived organoids do not contain the full mixture of cells or fully mimic tissue structure.
Is he up there rehearsing?
Adult stem cells cannot be used for every disorder, and it is dangerous to suggest this is the case. To close, I would like to reiterate that fetal tissue is critical for biomedical research to develop treatment for many human diseases. Alternatives cannot be proven to work unless they've been compared to fetal tissue. Without validation, patients are put at risk. Fetal tissue remains essential, and it is simply wrong to suggest otherwise. Thank you, and look forward to taking questions.
Thank you, Dr. Temple. I'm sure that you'll